Okay, good afternoon, everyone. Can you hear me?
Yes, we can.
Okay, thank you. Okay, good afternoon, everyone. Welcome to Breakout Room 4 of ISSAPA 2026. My name is Nurmaisa and I will be your moderator for this session. I am a PhD student in the International Doctoral Program in Agriculture at National Chung Ching University in Taiwan. Thank you all for joining us today. We will begin shortly. Before we start, I would like to ask our presenter to test the microphone and screen sharing. The first presenter, Ms. Alfian, could you please test your microphone?
Okay, hello.
Yes.
Can you hear me?
Yeah,
Okay.
thank you. Your microphone is working well. Could you please test your screen sharing?
Okay. Can you see my screen sharing?
Yes, thank you.
Okay.
Yeah, your screen is visible. And then the second presenter, they join in the on-site here. And then next, the third presenter, Miss Anjar, could you please test your microphone?
Hello?
Yes.
Okay.
Okay, thank you. Your microphone is working well. Could you please test your screen sharing?
Okay, wait a minute.
Yes.
Is it okay? Can you see my
Okay,
screen?
okay. Yeah, okay. It's visible. Okay, today we have two reactors for this session. First, Dr. Abdul Rahim, Dean of the Faculty Agriculture at Borneo Tarakan University in Indonesia. And then the second, the Miss Dit Hertzu, the from student International Master Program in Plant and Microbial Biology. Good afternoon, discusser, actor, presenter, participant, and guests. Welcome to the parallel symposium session of Breakout Room 4 Agroecology at ISAFE 2026. Before we begin, I would like to remind everyone to keep their microphone mute during the presentation. Each presenter will have 12 minutes for their presentation. I will give a reminder when there are 2 minutes remaining. Question and comment will be welcome during the discussion session after all presentations are completed. Thank you for your cooperation. Let's begin with our presentation first. Our presentation first, Miss Alfian Silvia Krisnasari. The title of the presentation is Nutraceutical and Functional Food Potential of Wild Edible and Medicinal Plants in East Kalimantan Forest for Stagnant Food and Wellness Security. Miss Alfian, you may start now.
Okay. Wait.
Miss Alfian, can you share your presentation? Okay, maybe Miss Alfian, they didn't have joined here. Okay, sorry. Maybe we can move the second presenter. For the second presenter is Mr. Sheng Yang Lee. The title of the presentation is Optimizing Black Soldier Fly, Nutritional Profile for Sustainable Feed Effect of Diet Potential Level of BSF Protein Context. Mr. Lee, you might start now. Yeah. Okay. You can start.
Okay. Thank you very much. Very good afternoon, Professor, my fellow speakers, and the remaining participants. I'm Sheng Yang, a master's student from the Department of Entomology inside Plant Interaction Lab. And today I'm very excited and honored to share with you my bachelor research on optimizing black soldier fly commercial illucens nutritional profile for the purpose of sustainable feed. And the main experiment is about the effect of dietary protein levels on the BSF protein contents. This work was conducted alongside my advisor, Professor Huang Xiaoyi of the same lab. Without further ado, let me start. As the global population expands, the pressures on the agriculture and global food supply chains intensify. The demand for sustainable animal and aquaculture feed has never been more urgent. Today, we will explore how we can optimize an insect-based solution to address this critical global challenge. It's not shared. I thought it was... When I restart. Oh, that's awkward. I restart the whole thing. Okay. A very good afternoon, Professor, my fellow speakers, and also remaining participants. Thank you for being here today. I'm Sheng Yang, a master's student from the Insect Plant Interaction Lab, Department of Entomology. And today I'm very excited and honored to share with you my bachelor research on optimizing black soldier fly commercial illucens nutritional profile for the purpose of sustainable feed. And the main experiment is about the effect of dietary protein levels on the protein contents of the black soldier fly. This work was conducted alongside my advisor, Professor Huang Xiaoyi of the same lab. Without further ado, let me start. As the global population expands, the pressures on the agriculture and global food supply chains intensify. The demand for sustainable animal and aquaculture feed has never been more urgent. Today, we will explore how we can optimize an insect-based solution to address this issue, this critical global challenge. To start, let's answer fundamental questions. Why the black soldier fly or Hermetia illucens? First and foremost, they are exceptionally rich in protein source. Depending on the life stage and feeding regime, black soldier fly meal can boast a crude protein range from 21.6% to 65.5%. It can also pack with essential amino acid, which is required for livestock growth and development. This makes them a highly viable alternative to traditional environmentally depleting protein source, such as fish meal, which can cause overfishing, and soybean meal, which can cause deforestation. These two are commonly used in aquaculture and livestock industry as their feed ingredients. Ultimately, utilizing BSF, black soldier fly, represent a circular economy approach, transforming organic waste into high-value nutrients. Our goal is to maximize this nutritional potential to ensure they serve as a predictable, high-quality, and sustainable protein source. This brings us to our core questions, research objective. While we know BSF larvae are excellent converters of organic materials, past literature tells us that their final nutritional profile heavily reflects the property of their feeding medium. Therefore, identifying precise feeding strategy is critical if we want to maximize and standardize their nutritional outputs for commercial feed formulations. Our investigation centered around one key question: how does diet protein level affect BSF protein contents? Based on traditional nutritional model, we formulate a straightforward hypothesis. As the diet protein levels increase, the BSF protein content should also increase. To test this hypothesis, we designed a structured 5-step experimental methodology. Firstly, with egg collection, we harvested fresh egg from our lab flash black soldier fly breeding colony. Second, with diet analysis, we do chemical analysis to run on the experimental diet to test out the protein content in the diets. Followed by larva rearing, larvae were introduced to specific controlled dietary environment. And also pre-pupa collections, we track the developmental process precisely and harvest the individual right as they transition into the pre-pupa stage. And lastly, BSF analysis. We will quantify the exact crude protein levels within the harvested insect tissue in the egg. Let's zoom into the timeline of the rearing process. Our experimental lifespan about 17 to 20 days after hatch. Egg collected will be left to hatch, and after the larva hatch, they will undergo a controlled feeding phase for around 10 days, stabilizing before entering the experimental feeding phase for a week. By day 20, the pre-pupa will be collected. For our diet formulations, we utilize a combination of soybean residue, wheat bran, and chick feed. Our control group consists strictly of wheat bran and chick feed. And to maintain a strictly scientific accuracy, each dietary treatment was conducted with 3 independent replicates. We formulated 6 distinct experimental diets to systematically vary the protein levels. We added soybean residue in graduals of 25 grams increment, ranging from 25 grams in diet 1, all the way up to 150 grams in diet 6. Concurrently, we reduced the wheat bran content proportionally to ensure that the total dry weight remained perfectly consistent across all treatments. Once the larvae reached the pre-pupa stage indicated by their darkening of their cuticles, they were collected, thoroughly washed, oven dried, and also crushed into powder for rigorous composition analysis. Then the samples will be weighed and prepared for analysis using a microcardia assay process to measure the resulting crude protein levels, and the result will be recorded for statistic analysis. For microcardia assay, 50 milligrams of the samples will be dried, and the pre-pupa powders will be subjected to high temperature acid digestions. This was followed by a color development using nasal reagents to form an orange complex. We then measure the light absorptance using a spectrophotometers under 460 nanometers. And against the standard curve, we can calculate the percentage of the nitrogen. Finally, we calculate the final crude protein percentage for each crude by using the conversion factors of times 6.25 to convert the nitrogen percentage to crude protein percentage. Now, let's dive into the data. This scatter plot illustrates the relationship between the diet crude protein on the x-axis and the resulting BSF crude protein percentage on the y-axis. When we look at a linear regression line, we notice a very weak positive slope. While the line mathematically suggests the higher protein diets may lead to a slight upward trend in the insect body, the R2's value is incredibly low. As you can see here, is 0.0169. Statistically speaking, our F value was 0.2751 with a P value much greater than 0.05. This means that the relationship between dietary protein and the final crude protein concentration in the prepupa was not statistically significant. To explore this further, we perform a one-way ANOVA to compare the mean across the dietary group. As you can see in the data tables and the corresponding blocks plot, the average crude protein contents is about 17.2% and to 17.6%. You will notice that the diet 4 shows a visible dip down to 14.97%. Upon close statistical examination, diet 4 was identified as a clear anomaly due to external reeling variations and was subsequently omitted from the main comparative analysis to prevent screwing up the whole biological conclusion. In terms of regression shown previously, our F value was 0.2751 with P value much greater than 0.05, meaning that they are not statistically significant. When evaluating the remaining group, the ANOVA confirmed no significant difference between the means with a P value greater than 0.1. This leads us to a fascinating conclusion. The variations in our tested dietary protein level did not significantly impact the final crude protein contents of the black soldier fly. Their protein composition remained remarkably stable. Our finding point towards several critical biological implications. Firstly, why didn't a higher protein diet use a higher protein insect? This suggests that the protein content in a black soldier fly is highly regulated and bounded by their genetic. Instead of dietary protein levels alone, factors such as substrate digestibility, whether how good it is, the specific amino acid profile, and also the overall larva genetic likely play a far more dominant role. Furthermore, developmental timing matters immensely, as illustrated by the infographic here. Maximum protein deposition and elimination happen during the intensive growth phase of the early feeding larva stage. By the time the insect reaches the prepupa stage, which is the metamorphosis preparation phase,
you have 2 minutes.
protein accumulation plateaus, energy usage shift from nutrition deposition to maintenance and building of the chitinous pupal casing, meaning that new dietary protein is no longer being actively stored as muscle or tissue protein. Lastly, a key limitation of this study was the relatively narrow range of the protein tested in our diets. A much wider extreme protein gradient might be required to force a homeostatic shift to their baseline. Looking forward, we aim to expand this research along 2 main pathways. Firstly, nutritionally, we plan to test on different organic matters, the parameters to measure other than protein should be lipids and carbohydrate alongside protein. We also want to retest our hypothesis using extreme protein heavy substrates and analyze the insect at multiple stages of their life cycle to map out the exact stage peak nutritional value is reached. Secondly, we are currently integrating advanced technology into the BSF insect farming. We are using AI-powered image recognition software to track black soldier fly larva activeness. By monitoring how quickly larvae respond to stimuli within a substrate, we can use computer vision to establish a correlationship between larva activity levels and organic waste conversion efficiency. This automated monitoring could revolutionize the commercial production scaling. In closing, I would like to express my deepest gratitude to my advisor, Professor Huang, and my lab seniors for their invaluable guidance throughout this project. Special appreciation goes to the Insect Plant Interaction Lab for providing the excellent facilities and equipment that make this work possible, and our university for providing the research grant to fund this study. If you would like to read our full report or examine our reference, please feel free to scan the QR code on the screen. With that, thank you very much for your time and attention. I'll now open the floor to any questions that you may have.
Oh, thank you very much, Mr. Lee, for your presentation. We will move to another presentation. Maybe I can move the first presentation for the Miss Alfian. Miss Alfian, can you
Okay.
hear me?
Yeah, yes, I can. I'm sorry.
Okay. Can you share for your PPT?
Okay, wait.
Okay. Okay, the first presenter is Miss Alfian Silvia Krisnasari. The title of her presentation is Nutraceutical and Functional Food Potential of With Edible and Medicinal Plants in East Kalimantan Forest for Strictness, Food and Wellness Security. Miss Alfian, you may start now. Oh, maybe she is missing again. Okay. We can move the another presenter for the third presenter, Miss Anjar Cahyaningtyas.
Okay.
Yeah, okay. Can you share your PPT?
All right.
Okay, our third presenter is Miss Anjar Cahyaningtyas. The title of her presentation is Plant Dynamics in Grazing Area Established on Abandoned Farm. Miss Anjar, you may start now.
Okay, thank you. Thank you, Ms. Nurmaisa, for the time for us to present our study. So maybe this is not kind of fancy study like the previous speaker, because this is our just simple experiment in our own garden. So our title is Forage Plant Dynamics in Grazing Areas Established on Abandoned Farms. So this is our background. So in our area in Yogyakarta, there are a lot of farm that is used for salak production. Salak is snack fruit. So in many area, the salak is become non-productive, and then some of the farmer transform the non-productive area into the forage grazing area. So the consideration is to support the sheep farming business, because in Yogyakarta also known as one of farming business area. And then why they transform it to forage grazing area, because it's less costly compared to another crop production. Then it's relatively easy compared to another community. Then what we did to our land, the first we conduct, so we use minimal soil management, because we want to support the regenerative agriculture. So here we only use shallow tillage, and then we apply manure, and in some of our experimental area, we apply the mycorrhizal fungi. Then after that, we apply or we sow the targeted forage species that usually used for sheep forage. But the problem is there are so many unwanted or non-target seedling that comes from the native soil in the post-salak production. Then this is the left picture is before planting, and then the right picture is the after planting. So we can see that there's a lot of plant species grown in here, and most of them is not our targeted plant. So the aims of our study is to observe the effect of arbusculum mycorrhizal fungi application to see the competition between the native and also targeted forage plant species in our experimental area. And then this is our research site. So we conduct our study in Sleman in Yogyakarta, and we partnering with one of sheep farmer over there. The name is Kandang Sheep Boss. And then, as I mentioned before, we use minimal land management. We clear the land, and after that, we conducting a minimal tillage and apply the manure and also mycorrhizal fungi, and we sow the seeds. So the targeted forage species that we sow as a seeds in our experimental area is Brahyaria. We have 2 Brahyaria species here, and also Setaria, Medicago sativa, and also Arachis pintoi. These 5 species is known as sheep forage in sheep farming, usually in the area. Then how we observe and also measure. So we observe the planting area after 1 month, and then we use a grid. We use 1.1 meter plots, and then we have in total 10 plots, and we identify the seedling growns in the area, and then we compare the growth of the target and also non-target plant species. Then this is one example of our grid. So this is the non-target species, and then also we identify the bare area and also the targeted species. So after we measure all of our plots, we found that almost of everything was actually non-target plants. But in the beginning, we found some of them are target species. Then from the biomass here, please see at the figure B, we found that in the mycorrhiza and also non-mycorrhizal field, we found in both of them, the trend is similar, but that is the non-target species is higher compared to the target species. But in mycorrhiza, it's not significant different, meaning that it's still comparable, the target species and also non-target species, meaning that the competition is not as high as in the non-mycorrhizal area. And this is maybe our, how to say, our most important findings. So actually, we check the dynamics or the difference between the coverage in each week. And here from the figure C, you can see the yellow color is the targeted species with mycorrhizal application. And then the green color, the circle code here is the mycorrhizal application, and then the triangle is non-mycorrhiza, and then the yellow color is the targeted species. Then the green color is non-target species. Here we can find that in the beginning of the study or first week after we sow the seeds, we found that the target species dominating the area, but then it's changed. In the second week, in the non-mycorrhizal area, the non-target species start to outgrow the target species. And then in the mycorrhizal field, we found that this condition, or when the non-target species outgrow the target species, it happens in the third week after sowing. Like this indicating that the mycorrhizal application may have implication, or it can affect the competition between the target and also non-target species in our study field. However, it only happened in the initial stage of the experiment, because after third week, it's also changed. Still the non-target species outgrow the target species. Then we also observe the dominant species of non-target plant. We asked the local farmers about the name of the species, and then the forage potential, are the farmer also use these species as forage or not. And we found that there are 5 dominant species. We found Cynandrella nodiflora, Ageratum conizoides, and Crasocephalum physalis, and also Digitaria. And among these 5 dominant species, the 4 species are having the potential as forage for sheep farming. And this is the picture of the plant species. Then come to the conclusion. So we conclude that overall in our study, the species emerged from the soil seed bank, or the native plant species from the seed bank outgrow the target species. And then the second one is the inoculation of arbuscular mycorrhizal fungi may enhance the early competitive performance of our target species relative to the non-target species, even though it's only happened in the initial study. Then further, maybe we would like to study about the palatability or quality of the non-target species as a fodder, and also their significance in pasture management. So here, let me introduce our team. Here we have a lecturer from Universitas Pembangunan Nasional Veteran Yogyakarta, and also we collaborate with the local farmer, and they practicing the integrated farming management in the area, particularly in Sleman and also Klaten area in Indonesia. And that's all from me. Thank you very much.
Okay, thank you very much, Miss Anjar, for your presentation. Okay, I can check again for the Miss Silvia in the main room. Wait a minute. Okay, I think the Miss Silvia, they cannot join today. Okay, thank you for all presenter for sharing your work today. We will now move off the reactor session. The first one, I would like to invite Professor Dr. Abdurrahim to share his comment and suggestion. For the Professor Dr. Abdurrahim, please go ahead.
Thank you very much, Ms. Nurmaisa. I'm not asked, but I want to more explain from the speaker. The first speaker is Lee Sheng Yang, research about the black soldier fly. I want to ask you about your what's the meaning background or consideration or judgment. Why you choose 6D3 protein, especially from soybean? What's the meaning residue, and what the meaning of the soybean residue in your research? Maybe you more explain about that. And then why you only analyze the pre-pupa, and then what do you think about your sample size per dietary group? I think you have 6 dietary percent, but you only have 3 group it's compatible for your analyze. Your analyze using what's the meaning correlation and what's the meaning analysis of variance. And finally, your research has no significant effect on the 3BSF. This is I think I need more explain why no significant. This is problem because your statistical analyze or you maybe miss to choose dietary protein for the BSF. And also I want to ask why you choose method kids DAL for analyze crude protein. Why you not choose near infrared spectroscopy? I think it's a different method for the analyze crude protein. And yeah, I think it's enough. And what this is the farmer in my country, especially livestock, it's not to what's the meaning like BSF for BSF for feed for the livestock because BSF have so many chitin, so it's difficult to digestive of livestock. So why you choose BSF as potential as a feed on the livestock? Maybe this enough for my question.
Okay, Mr. Sheng Yang, can you?
Lee.
Okay, thank you, Professor. Okay, let me confirm a few questions first. So you're asking me why do I measure only the protein content? Why do I use microdigest assay to measure the protein content? And why did I use food waste and I choose soybean residue instead? Am I right?
Okay, that's right.
Okay. Okay, well, the first part for protein contents, because my goal is for sustainable feed. And for feed, the most important thing in most case is the protein content. So that's why my first major focus is the protein. In terms of why do I use microdigest assay is because currently that's the only available methods that is available in the lab. So that's why I can only use that to measure protein. But if let's say I have the other methods, I would definitely want to try it out. And in terms of why didn't I use waste such as manuals for the experiment is because in Taiwan itself, if we are going to use manual to feed the black soldier fly, there's an issue that we cannot use the black soldier fly to feed the pig, and because the pig is going to be fed to us. So there's a regulations about it that is not ready for us to use manual to be into the system. So currently that's the limitation. Then is why do I use soybean residue is because that is a mistake on my part. Because when I doing this research, my first thing I'm looking for is protein source. So soybean meal have been one of the main ingredients, but one word there's a lot of different. One is meal, one is residue. Residue are byproducts of after tofu or soy milk production. So even though it's from the same source, but the protein source can be very, very differently. So for soybean meal, it can go up to 40+%, but for soybean residue, when I measured the protein content itself, it's only about 9 to 10%. So it's actually not really that high. So that's the mistake on my part and also the flaw in my experimental design that I need to improve. That's why currently in my master research, I also trying to edit that part. Yeah, that's all.
The last one maybe about your analyze statistic analyze. Why you choose the regression and of variance for your statistic analysis?
You mean why do I only use one-way ANOVA and also regression?
Yeah, and
Currently that is the most straightforward method to tell whether there's a significant difference between the two. That's the first thing. And also are only measuring protein, so there's not much variables that I think I need to consider. So that's the first thing. Yeah.
Okay, thank you. Please.
Okay, thank you, Professor Dr. Abdul Rahim. Maybe you can ask the another presenter for the Miss Anjar.
Me again? Not the other participants?
Miss Anjar, for the Miss
Oh,
Anjar.
forage plant dynamics?
Yes.
Oh, okay. Okay. Same like a previous speaker, I just know what are your more background. Choose the abandoned snake fruit, Salaka in Yogyakarta. How large potential, the how big potential land abandoned snake fruit, Salaka. And also your method using 10 plots, and each plot 1 meter square. This is representative uh, for the abundant snake fruit in Yogyakarta, and you always use terminology about non-target plant. Yeah, but I look the species in agriculture faculty. Maybe you mean non-target is wheat. Yeah, wheat or gulma in bahasa. Yeah. Uh, why you spirit non-target, uh, target, and what's bear? Yes, bear. Yeah, for the your analyze, and then you measure too. This is about methodology. Presentation coverage. Three target or three object. How to measure the target, non-target, and barrier, and uh, you have a recommendation to harvest of grass around the third week. Yeah, please more explain why you recommendation that, and then yeah, what is the ecological or economic rationale for recommending your research in your research. Maybe that's all. Back your moderator.
Okay. Miss Anjar, can you answer the question?
Okay. Thank you, Prof. Abdul, for your questions. So the first is about why or how many is the abandoned farm in Sleman, right? So actually, I don't have the intake data about that, but it's increasing right now. It's still increasing because there's a lot of people that they grow old and they don't have descendants to continue the salak plantation, so they just abandon the forest. And I think that will not be productive if we do not manage the salak, because salak is need so many input. Usually, like they use a lot of fertilizer and etc. But when they abandon the area, maybe it will just become so nothing can we get from the area. But when they transform to ladang gembala or the forage grazing area, it will increase the amount of the forage for the sheep they they manage in that area. Then the second one is the representativeness of my samples. So actually, our study site is not that wide. So I think it's only around 1 hectare, something like that. And then we choose the sample like in we choose the plot location that can represent the total area. So I think it can represent the area. And then when we check the species in each plot, it's quite the same in all the plots. So I think it's can represent the total area. And then why I use the terminology target and non-target, because actually I plan the target. So here we have target species, and maybe because why I use the term target, because I don't want to say it's crop, because yeah, I just want to distinguish the target and non-target, because the target one is the grass species that usually use for forage, and then the non-target is, yes, the gulma or uh, the other species other than the one that I plant in that area. So I just want to distinguish between the one that I sow and the one that grow from the native soil, from the seed bank, soil seed bank. Then how do I measure the coverage of the target species, non-target species, and also the bare area? So I use grid. I use a small grid, one and 1 meter plot, small plot, and then I divide into 4 quadrant, and from that I can actually it's semi-qualitative. So I measure that area from the 1 by 1 plots. So from there I can check this area is how many percent from my plot, and this target species is how many percent from my plot, something like that.
Okay, thank you, Miss.
The economic and ecological rationale for recommendation. Your recommendation to harvest of
Okay.
those.
Oh, sorry. Why I recommend that, because I see that when I apply mycorrhiza, my target plants or the seeds that I sow, they can uh more they have higher competitiveness compared to the gulma or the non-target species or the weeds. So I think in the third week we can get more biomass from the target species or the grass that I sow. And then uh, economically, if the biomass is higher, I think the farmer can get more forage for their livestock. So I think that's why I recommend that they should harvest the plants in the third week, like that, Professor.
Thank you.
Okay.
Okay, Miss Nursing. Okay.
Okay, thank you. Thank you, Professor Dr. Abdurrahim, for your valuable comment and suggestion. And next, I would like to invite Ms. Tiet Hertzu to share her question and the comment. Ms. Tiet Hertzu, please go ahead.
Um, maybe the party Indonesia asks Mr. Sanya.
Yeah, you can ask first Mr. Sheng Yang, and then you can
Yeah.
ask the Miss. Ms. Anjar.
My question is that since you presented one way innover show no significant difference across dietary groups, how do you rule out that the development state itself, rather than the diet, is the primary driver of protein content stabilization?
Okay, for this, right, this will be because of the distinct sociological roles of each developmental stage. When we break down into the life cycles of black soldier fly, the main two phases will be the larva phase and also the pupa phase. The pre-pupa phase is the phase in between these two. And during the larva phase, this is the active feeding and growth phase whereby proteins is accumulated and from the food they eat. When they transition into the pre-pupa phase, it's a sort of like a metamorphosis preparation phase. So at this point, they stop feeding, so the protein levels will stop accumulating and it will start dropping because they need to form a chitin in their cuticles to form the pupa casing. So that's why when you tested the pre-pupa protein level, it will be lower compared to if you measured in the larva phase. This is also many research have also shown that the larva phase had a higher protein level than the pre-pupa phase. So that's why during this phase itself, you can rule out the dietary variation because majority of the changes is happening because of the psychological homeostasis tied to the life cycle stage. Yeah. Do I need to answer the professor's question?
No, I think so.
Okay.
Okay, maybe you can ask again. So, yeah.
Okay, another question is to Mix Anjar Cahyaningtyas from Indonesia. My question is that you presented about the forage plants dynamics in grazing areas, and I want to know that the competitive advantage of AM fungi inoculated target forage appears short-lived. What mechanisms might explain why non-target species eventually outcompete the target forage despite inoculation?
Okay, thank you, Miss Su, for your question. So, why the non-target outcompete the target? I think because the target species is actually not native in that area, and we choose that species. Our target species is because it's usually used for the grazing area, but actually it's not native in our area in Sleman. But the non-target species actually is originally for soil seed banks. So that's why maybe they already adapted to the area. So that's why maybe even though we put the mycorrhizal fungi, we inoculate our target with the mycorrhiza, maybe they still outcompete with the local species. Maybe that's why. Maybe because the adaptiveness of the species is different. The one is from outside the community, outside the area, and the other one is from local area. Maybe something like that.
Thank you for your answer.
Okay, thank you, Ms. Jit Harsu, for your question and comment. Now we will know when the floor for question and discussion. If you would like to ask a question, please raise your hand or type your question in the chat box. Okay, I can check in the chat box. Oh, okay. I have the question from the Mr. Pratoyo from Indonesia to Mr. Lee Sheng Yang. Your research did not show the potential of BSF as a potential source. What are the main potential target of raising black soldier fly as a source of fish and livestock feed? Okay.
Okay. Good afternoon, Professor. So, to answer your question, yes, this research indeed never showed the potentials of black soldier fly. That's a mistake on my part. But other research has shown that actually the potential is yet to be really fully discovered. So I still believe in potential in black soldier fly. So currently, there's a few black soldier fly is commonly used as a supplement or instead of full black soldier fly diet. So they are mainly used more of supplements. So for example, in Taiwan, some small scale chicken farm, they will use black soldier fly to feed on them for them to eat. And in Singapore, based on what I know, black soldier fly is also raised to feed small scale fish farm also. So fish farm, chicken is a few common targets that black soldier fly is constantly used. In the pet industry, black soldier fly is also a racing star because it's also currently being used to feed such as reptiles, oriental fish also. Yeah, to answer your questions.
Yeah, Mr. Pratoyo.
Yes, thank you for the answer.
Okay, thank you. Okay, maybe one presenter they can back again. This is Miss Alfian Silvia Krisnasari. You can hear me? Hello, Miss Alfian Silvia Krisnasari. Can you hear me?
Hello, Miss Alfian Silvia. Can you hear us?
I think no one. I don't know, maybe miss again. Yeah. Yeah, maybe. Okay. Thank you for all. We have time for the final question. Thank you for our reactor, presenter, and participant for today's discussion and valuable contribution. On behalf of ISAFE 2026, I would like to thank everyone for joining this session. This concludes the parallel symposium session in breakout room 4. Before returning to the main room, certificate will be presented to our presenters and the reactor. I kindly ask everyone to stay for a moment. I can share the certificate. The certificate for the presenter Sheng Yang Lee, the title Optimizing Black Soldier Fly, Nutritional Profile for Sustainable Feed Effect of their Potential Level of BSF Protein Contents. Designed the Dr. Silvia Wang and Director International Doctoral Program in Agriculture, National Chung Sing University. And then the design Dr. Ji Feng Chen, Dean College of Agriculture and Natural Researchers, National Chung Sing University. You can take photo.
You may want to repeat because the
Yeah.
video is not on. That's not. What? Okay.
It's okay.
So,
Okay, next, this is the certificate for the presenter, Ms. Anjar Cahyaningtyas, the title, The Forage Plant Dynamic in Grazing Areas Established on Abandoned Farms. And the signed the Dr. Xiao Huang, Director, International Doctoral Program in Agriculture, National Chung Sing University. And then the signed Dr. Ji Feng Chen, Dean College of the Agriculture and Natural Researchers, National Chung Sing University. Okay, oh, sorry, I can read the chat. So you can please. For the Miss Alfian, you want to present? Can you hear me?
She's really has surgery. Maybe she's in hospital.
Oh, okay, okay.
So she is back.
Yeah.
We have no time.
Yeah, we don't have time again. We can back to the main room. Okay, congratulations to all the presenter and reactor. We will pause off for the moment to allow everyone to take the screenshot their certificate. And then please feel free to take your screenshot now. And participants may not return to my room. The closing ceremony on Instape 2026. Thank you very much for joining us today. Have a wonderful afternoon. See you in the main room. Thank you for the Professor Dr. Abdul Rahim, and then the presenter for the Sheng Yang and Ms. Anjar Cahyaningtyas, and then Mr. Pratoya from the Indonesia. And then can you back to the main room? Thank you for today.
Thank you.
Thank you.